Okay, so bacteria can use to be transferred DNA. So we just looked at how DNA can be used in crime scene investigations, paternity, in establishing evolutionary um, linkages, but we also want to, we can use DNA to transform a genetic code. All right. And one of the ways that we do that is through the use of bacteria. So we need to learn a couple of things about bacteria before we get started. First of all, bacteria use three mechanisms to move genes from cell to cell. Transformation, which is the uptake of DNA from outside in the environment somewhere. It just sees floating around pieces of DNA and sucks it in. Transduction is a gene transfer by phages. Phages are viruses that attack bacteria, and conjugation, and that is the transfer of DNA from one bacteria to another. All right. Now let's look at pictures of what those look like. Oh my goodness. Let's look at pictures. So DNA, there's DNA fragment just hanging out. Maybe there was a bacteria that got killed and its DNA got chopped up and pieces of its DNA are just floating about. Or maybe we put the DNA in there, right? Scientists put the DNA in there. The DNA is just going to be brought into the cell. Okay, so that's transformation. If it uses a virus called a phage, here's the virus, then we can use that virus to inject the DNA, or the viruses can attach and the bacteria can attach, excuse me. And one bacteria is going to give genetic information to the other. It will not go in the opposite direction. It only goes in one direction. All right. Now, once the donated DNA enters the bacteria, it can cross over and integrate itself into the DNA of the bacteria. Then we call it a recombinant DNA or recombinant chromosome. Recombined is basically what that means. We recombined. We combined two different pieces of DNA from two different organisms. And we call that a recombinant chromosome. We're going to find that we have recombinant genes. We're going to find that we have recombinant organisms. So typically we use E. coli, which is just a really great basic bacteria, to carry out conjugation is due to something called an F factor. And I don't necessarily care that you know F factor, but I do care that you know it is a plasmid. And a plasmid is a small circular piece of DNA that is separate from the actual bacterial chromosome, which is a large circular piece. Okay? So I'm going to leave you to answer the checkpoint questions. Make sure that you are answering these checkpoint questions, as that is how your notes will be graded. If the F factor is an entire plasmid, see, this is the bacterial chromosome. A plasmid is just a circular piece of extra DNA. Okay? This is the method we're going to talk about most. This is the one I care about. This is the one that's in your notes. A plasmid is a separate piece of DNA, separate from the bacterial chromosome. The F factor is going to replicate separately from the genome of the bacteria. That plasmid is going to then be transferred in its entirety to the bacteria. So now the donor, the cell, now has a plasmid here. This one has a plasmid here. That's conjugation. So how do we use this? All right. We use this in gene cloning. This is one application of biotechnology. We take these plasmids, we can create these plasmids, and then we can actually get the plasmids to be inserted into the genome. And when we do that, they're called recombinant DNA. Okay? Recombined DNA. We've recombined DNA in the plasmids. We can insert these into the bacteria. And then the recombinant bacteria can multiply into a clone. And a clone, we think sometimes, we can clone plants and animals, but really that starts with being able to clone DNA. So let's see what this looks like. All right, let's start up here. We isolate a plasmid, okay? And then we isolate whatever gene, a cell with the gene that we want. So let's say we are going to use insulin as a really good, insulin's a really good thing. So we can take the genes for insulin, cut them out of here, take the plasmid and cut it out of here. We are then going to cut both of those DNAs, both of that DNA with the exact same enzyme. And then what happens is we can actually stick them together. Because we cut them with the exact same enzyme, they're going to leave this sticky edge. Well, sometimes they leave a blunt edge, but we'll talk about that in a minute. And then they can be put, the actual piece of DNA that you want can be put into the plasmid like this. We are then going to take DNA ligase, which if you remember DNA ligase, this is what put together our Okazaki fragments. We put together, we use DNA ligase, we join them, and now we have a plasmid with our gene of interest. This is now called a recombinant DNA or recombinant plasmid. We're then going to put this gene into the bacteria. The bacteria is going to reproduce. The bacteria is going to then create the proteins. We can then put the genes in other organisms, or we can harvest the proteins. So in this case, we would want to harvest that insulin protein. This is the insulin that is going to be used to give to diabetics. All right? So look at 12.1b. What is the vector? So if you look at 12.1b, you're looking for the vector. Vector is going to be the bacterial plasmid.
We can use the techniques of DNA technology to recombine and copy genes. A restriction enzyme is used to cut open a plasmid, a small circular DNA molecule obtained from a bacterium. The plasmid serves as a cloning vector. The restriction enzyme cuts only at a certain DNA base sequence, called a restriction site. The same enzyme is used to cut up DNA obtained from a human cell. The human gene must be cloned to produce copies for use or study. A bacterium is induced to take up the recombinant plasmid from the surrounding solution in a process called transformation. This isn't as clear-cut as it sounds. Some bacteria do not take up the plasmid, and some take up plasmids whose LAC-Z genes lack the inserted human DNA. A bacterium is placed on a growth medium. It replicates the plasmid at the same time as its own DNA. Its descendants form a clone of bacteria that all have copies of the recombinant plasmid. The ampicillin in the medium limits growth only to plasmid-containing cells. But which cells have plasmids with a human gene? Bacteria without human genes have intact LAC-Z genes. This allows them to break down a substance in the medium that stains their colonies blue. Thus, the bacteria with human genes are in the colorless colonies.
All right, so how do we cut the DNA? We use restriction enzymes. So we cut the DNA in specific places, forming restriction fragments. We then use DNA ligase to paste DNA fragments together. Okay? So those fragments have sticky ends. Okay? Sticky ends, I'm going to show you, are when we cut, so this is the restriction site. This is where the restriction enzyme is going to cut, right here at this particular short tandem repeat, right here. And it leaves an edge so that when this gene of interest is taken in and it is added to, it will fit just right. The AATTC will fit right next to the G. So it's almost like a locking end, not really a sticky end, but that's what we call a sticky end. So that is the answer to your checkpoint question. You're going to want to put that in. Once we match them up, they will kind of match themselves up because they will have the correct stickiness, essentially. And then DNA ligase is going to come by and stick them together. All right?
Restriction enzymes are enzymes isolated from bacteria that recognize specific sequences in DNA and then cut the DNA to produce fragments called restriction fragments. Different restriction enzymes recognize and cut different sequences of DNA. This restriction enzyme recognizes the DNA sequence GAATTC. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence AATT. This restriction enzyme recognizes the DNA sequence CTGCAG. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence ACGT. This restriction enzyme recognizes the DNA sequence CCCGGG. It cuts the DNA strands between the bases C and G. The resulting DNA fragments have no single-stranded sticky ends.
All right. We can also add a probe on the sequence. Now, why would we want to be able to do that? Well, we can add a radioactive probe so that we can find the ones that actually clone, because some bacteria is just not going to take up your cloning plasmid. It's just not going to, okay? So when we want to just get out the ones that we found, we can put a nucleic acid marker on it. We can also put an antibiotic marker. So if we put an antibiotic for, say, tetracycline, and then we put it in a tetracycline medium, then that means the only bacteria that will grow are the ones that have the plasmid with the tetracycline resistance, okay? All right, and this is an example of a radioactive tag, and that's pictures in your notes. Okay, so all this technology is great, but what can we use it for? All right, so they have developed this thing called CRISPR-Cas9. You're going to do a worksheet on it, and it is literally the coolest. What it does is it actually allows you to target a specific gene in a cell that is living and either remove the gene or edit the gene, okay? So here's how it works. It's so cool. All right, here's how it works. We have this Cas9 protein, and it has a guided RNA. And the Cas9 guide binds to its target sequence, okay? So the Cas9, here it is. It's got a loop right here, and it's going to bind to the guide. Or the guide is going to bind to the DNA, okay? The Cas9 is then going to cut the DNA. All right? So this is the resulting cut gene. You can then insert a normal copy. This is revolutionary. They can cure all kinds of genetic diseases. Now, this isn't going to work if you have a bacterial infection or a virus, but if you have a genetic disease, a disease caused by a problem with your DNA, okay, an inherited disease, or a genetic mutation caused by something else, you can use this Cas9 to actually edit and change your genetic information. So we'll be learning more about that later as well.